Welcome to Library Seminars, NOAA Central Library's virtual stage for the presentation of research and ideas that reflect the National Oceanic and Atmospheric Administration's mission. Today's presentation is NOAA's Cooperative Institute for Ocean Exploration, Research and Technology, linking exploration to conservation. Dr. Mashkor Malik, Division Chief of Science and Technology for NOAA Ocean Exploration, will introduce our speaker, Dr. Joshua Voss. Before I turn this webinar over to our speakers, here's some information for our live audience. We highly encourage you to ask questions, which the speaker will address at the end of his presentation. So please type them in the chat box throughout the seminar as they come to you. You'll find the questions chat box in the control panel. All audience members are muted and off camera. And finally, this webinar is being recorded and will be available on the NOAA Central Library YouTube channel later today. I will put the link to the channel in the chat box so that you can share it with others who may be interested in today's topic. So without further ado, the mic is yours, Dr. Malik.
Thank you, Lisa. Really happy to be here, and it's my unique honor to introduce Dr. Josh Voss. He is the Executive Director of NOAA's Cooperative Institute for Ocean Exploration, Research and Technology, or otherwise called as CIOERT, and an Associate Research Professor at Florida University Harbor Branch Oceanographic Institute. He grew up in Central Florida and attended Elon University in North Carolina and completed a bachelor's in biology along with minors in philosophy and chemistry. So quite a varied background. He earned his Ph.D. in biological sciences at Florida International University in Miami and was a member of the marine science faculty at Eckerd College in St. Petersburg before joining Florida University Harbor Branch, where he took over the role of CIOERT director from Shirley Paponi. His primary areas of interest include shallow and mesophotic coral reef ecology, coral health and disease, molecular ecology, marine conservation and management. And through Harbor Branch, Robertson Coral Reef Program, and CIOERT, he works to discover, characterize, and protect coral reef ecosystems. So Dr. Voss is a certified technical diver and scuba instructor who has completed over 1,500 scientific dives and led more than 35 scientific expeditions, primarily in Bahamas, Florida Keys, and Gulf. So the Cooperative Institute for Ocean Exploration, Research and Technology is led by Harbor Branch Oceanographic Institute at Florida Atlantic University, but there are other partners also, including University of North Carolina, Wilmington. And CIOERT mission is to apply disciplined innovation to continually improve, extend, and fortify NOAA's exploration, research, and operational capabilities. I'm also happy to report, like this, over the last 12 years, CIOERT has explored around 900 sites, covering over about 2,000 kilometers, and collected around 1,500 samples and archived around 100,000 research-grade images and published dozens of articles. So with that, I will pass it on to Dr. Josh.
Thank you very much, Dr. Malik. I appreciate it. And thank you, Lisa, as well, for coordinating and setting everything up. Welcome to those of you that are tuning in today, and for those of you that will see this later on. Please feel free to reach out with questions, further information, etc. I'll share my contact information here at the end. And we always love to hear from interested colleagues and other interested members of the general public about what they think of our work and maybe how we can share our information with them or what we can do better in the future. So please feel free to reach out. I want to dedicate this presentation to Dr. Natalie Villette-Silver. Many of you, I hope, had the pleasure of knowing her. She worked for NOAA for around 3 decades, and sadly, we lost her earlier this summer. Natalie was our science coordinator for CIOERT for several years and became a good confidant, friend, and always brought a very kind of broad, big perspective about what we can do to make our science more impactful. And she'll be greatly missed, but I appreciate all of her contributions, and much of what you'll see here today was inspired by or bounced off of Natalie at some point. Cooperative institutes typically are enormous, and CIOERT is one of the smaller cooperative institutes overall. That said, we still have a huge number of people that contribute to these projects. CIOERT has been based at FAU Harbor Branch here at Fort Pierce, Florida, kind of the southeast section of Florida. But we have two strong partner universities as well, University of North Carolina, Wilmington and University of Miami. We've also partnered with SRI International, who was a contributing partner, particularly in the early phases of CIOERT. And then as CIOERT began to develop and as we started to really try to meet NOAA, OER, and OAR missions, we also recognized that there was this huge opportunity to work more directly with sanctuaries. So many sanctuary partners, particularly Florida Keys National Marine Sanctuary and Flower Garden Banks National Marine Sanctuary, have been really integral and pivotal throughout the entire process. The other aspect of our cooperative institute is that while the bulk of our research support came through the Ocean Exploration Office, we also have several other projects that have been funded by other parts of NOAA, principally NCOS. In terms of the FAU team, the Harbor Branch team that has contributed to this work, over the years has been various projects that have come and gone, but there's been some that have been consistent throughout the entire history of CIOERT. And there's two people in particular I'd really like to recognize. First is Shirley Pomponi, our former executive director. She's got things off running quite quickly and aggressively, and we had several years of multiple successes. And then I was asked to take on this role in 2018. So for the past 5 years, I've been doing my best to fill those rather small but formidable shoes. For those of you that know Shirley, she's rather petite. The other person that I wanted to particularly call out is John Reed. John Reed is going to be retiring from FAU Harbor Branch here in just a few months, but he has been a huge contributor and leader on many of the projects that I'm going to discuss today. He is the consummate ocean explorer and advocate for conservation. I also am inspired by him and his unwillingness to back down and his resolute nature about making sure that we do what's right in terms of conservation. Many other PIs have contributed to this as well, or co-investigators, I should say, as well as partners at other institutions. So our largest partner has been UNC Wilmington. Dan Baden led the contingent at UNC Wilmington for a number of years as well. Also, when we're doing this kind of expedition-based work, we can't do it without the support of really skilled and qualified ship crew and ROV and submersible teams. So both the crews of the Manta, the Walton Smith at Miami, formerly the Seward Johnson at Harbor Branch, as well as the ROV team at UNCW and the former submersible team here at Harbor Branch, all made great contributions to some of the work I'm going to talk about today. And then, of course, we've had amazing support and collaboration and partnership with NOAA throughout the Cooperative Institute. That's really the hallmark of all cooperative institutes, is this partnership that allows us to be flexible, adaptive, and help to meet NOAA's needs. What's unique about CIOERT is that most cooperative institutes are co-located with a laboratory of some kind, a NOAA laboratory of some kind. Instead, CIOERT was directly aligned with the Office of Ocean Exploration. So for several years, Alan Leonardi was the director of OER and someone who we work with quite closely. Natalie, Steve Hammond, Phil, Casey, Kelly, Frank, and earlier on, John McDonough and Gene Smith and Yvette have all been really instrumental in our success and our ability to be nimble and react to what is needed in the NOAA scientific community and NOAA mission. And then some of the work that I'm going to present today has also had really strong contributions from graduate students on my team. So I wanted to acknowledge those, and all those indicated here with asterisks are ones that have completed their degree, mostly working on CIOERT activities and moved on in their scientific careers. And all of them have stayed within science. Several of them now work for NOAA. So a brief kind of history of our Cooperative Institute. The agreement was awarded in 2009. We really honestly kind of got rolling in about 2010. In 2014, we completed a mid-cycle review where we got an outstanding review, and therefore renewed for another 5 years. Although we were allocated over $40 million in funding in terms of the amount we actually received, it was closer to about 18.4 million net, and many of you know how allocations versus expenditures actually go. But we've also been quite successful in being able to leverage those funds for additional support from other areas of NOAA like NCOS, NIH, NSF, and state funding here in Florida as well. So we've been able to leverage and get about just over 7 million that directly complements the CIOERT projects that we started with. We have 33 CIOERT investigators across 6 different institutions, and we've had a great run of 13 years, but we've just completed the end of our no-cost extension. So CIOERT is sunsetting, as they call it. That doesn't mean that the work we're doing is ending, but the Cooperative Institute framework that we're currently working under will change. So CIOERT has really been focused on 4 major theme areas. The first is getting out there and doing active, crazy exploration, looking for new targets, particularly trying to find places that we don't know much about yet. Many of those places that are relatively high value in terms of ecosystem function or ecosystem services and economic value often tend to be coral reef ecosystems. And so our second theme area, as directed by the FFO, was to focus on vulnerable coral and sponge ecosystems and try to understand and protect them more effectively. We've also been tasked with developing new ways to do exploration, advanced underwater technologies that can advance either the pace or the scale or the scope of what we're looking at in the ocean. And then lastly, the one that makes perfect sense for us as a university and is also near and dear to my heart as someone who enjoys teaching and enjoys mentoring graduate students, we're tasked with training the next generation workforce, trying to help to bolster the amount of expertise and experience that's available in graduate students and undergraduates so that when they're moving into the field, they'll be better positioned to take important roles within NOAA and adjacent agencies. So I'm going to reframe kind of those same 4 big ideas in 4 different sections of this presentation. I'm going to talk a bit about that exploration part, but I'm going to do kind of the exploration with the coral and sponge work together. And then I'm going to separately talk about some of the work we've done regarding biotechnology and development of therapeutics or pharmaceuticals for marine sponges. I'm going to talk quite a bit about some of the tech development that we've done, as well as aspects of processing and image work that have taken off as well. And then finish up talking about our education impacts and future directions for the research we have going. So let's start here with the exploration and conservation of deep and mesophotic coral habitats. So over the past 12 years, we've been able to conduct 46 different research expeditions. The vast majority of these have been on research vessels offshore. A few of them have been land-based in the Florida Keys or Mesoamerica, but the vast majority are on ships, be they NOAA ships or university ships within our group or our ship. And we focus primarily on trying to find these underdescribed or undescribed mesophotic coral reef ecosystems, these areas that we know are ecologically interesting and important, but are not well characterized and have not been discovered in many places where we expect they might be. In these areas, we focused not just on characterization and discovery, but also looking at new examples of what may be there. That's either new species to science or new species records in particular areas where they hadn't been recorded yet, and identifying fisheries resources so that they can be managed more effectively, or at least included in the management planning for the relevant agency that's responsible for those regions. And then our big goal is really to take the data and information that we're generating and make sure that that's getting to conservation and management goals. And a huge part of that means that we have to work ahead of time before we even scope out the entire plan, partnering directly with sanctuaries or fisheries management councils, and knowing what their needs are at the outset of these projects has allowed us to tailor and target our exploration and expeditions to meet the needs and the data requirements that they need to not only have information, but actionable information, something that they can make a management decision or recommendation on. And that's been a huge focus. I'm going to highlight a lot of that today. So we've been able to use a multitude of amazing techniques, toys, methods, if you will. We rely heavily on multi-beam bathymetry. And then we often, after we've identified potential high quality targets, our next best tool is typically ROVs to verify that those targets are high value and to start to run surveys where we can understand that habitat. In some instances, we've also deployed surface gliders and been able to utilize human submersibles as well. But the vast majority of the work I'm going to talk about today was work where we started off with the ROV, and then where it made sense, we would send in teams of technical divers as well. And ROVs are excellent at doing surveys. Tech divers are still better at identifying small phenomena and much more efficient at collecting samples per unit time than ROVs. And I'll talk a little bit about some other technologies that we've developed as well, like our Mesophotic sensing platform. So my first big take-home message for all of you today is that essentially everywhere we went hunting for Mesophotic reefs across much of Florida, Cuba, Mesoamerica, and the Northwest Gulf of Mexico, and Southern Gulf of Mexico, we found them. Mesophotic reefs are extensive across our region. They're relatively healthy in comparison to shallow reefs throughout this zone, and they're absolutely prolific. And if you look at the relative available habitat that could be potential Mesophotic reef versus shallow reef, they dwarf the amount of shallow reef area in this region of the world. I think that there's still quite a bit out there to be explored and discovered, and certainly areas that we've only gotten a first look at and don't know the whole story for yet. So I'm going to go through and highlight some of these areas that we particularly focused on, in particular those that have been success stories in terms of taking the data and information that we've gathered and resulting in a positive management outcome of some kind. So I was thrilled when we started our Cooperative Institute because it finally gave me the impetus to do something I'd wanted to do since early in grad school, which is work at Flower Garden Banks National Marine Sanctuary. If you ever get a chance, I encourage you to go there. It's about 100 miles off the coast of Texas, and not at all the place where most of us would initially think that coral reefs are prolific, but they're arguably some of the highest coral cover reefs in US waters, at least Atlantic and Caribbean US waters. So initially we wanted to focus really on benthic habitat characterization, help the sanctuary to develop more defined habitat characterization maps so they understood what was within their sanctuary and high value targets outside of their sanctuary, including new species records for the region. Using a bunch of these stratified survey approaches across several different habitats, we worked with members of NCOS and other partners to help generate maps and generate data, and perhaps more important than all of those, generate stories and videos that compelled people to care more about this region. And so I'm really happy to report that in 2021, the sanctuary expanded it from 3 initial banks that have been protected to 17 banks in total now. And that represents a roughly 300% increase in the total amount of MPA area that's protected within Flower Garden Banks National Marine Sanctuary. Working out there also gave us the opportunity to develop a closed circuit rebreather technical dive team here at FAU Harbor Branch. When you're that far offshore, you don't want to have to bring a whole lot of helium, so rebreathers are absolutely the way to go. And we've now been able to use that team and use this approach in several different areas in Florida, Bonaire, etc. We also were able to conduct a mission where we did the first test of the Global Foundation for Ocean Exploration's flyaway telepresence system. So most of you are aware that GFOE runs the instrumentation on Okeanos Explorer, and they're the ones that are responsible for making sure that we get those amazing high quality image videos, particularly those of us that are streaming them through an i2 service with 4K. So this was a very different kind of approach where instead of massive infrastructure on a dedicated ship, they drove a container from Connecticut down to Texas and were able to unload and set up the same kind of live telepresence system on the back of a ship, and it was incredibly successful. To give you an idea of what some of these images look like during that cruise, we were able to literally have Sanctuary Advisory Council members watching live in Galveston and in Houston while we were conducting some of the research operations in Flower Garden Banks. We focused on two things during this mission. First, to demonstrate the capability of the instrumentation. Secondly, to do some follow-up surveys to look for changes in these habitats. And then also, thirdly, I should say, we were looking specifically for evidence of coral spawning on Mesophotic reefs, and we found it. It was fantastic. So we've known about Flower Garden Banks, and we've known about some of these other banks in the Northwest Gulf of Mexico. When the opportunity arose for us to also expand this same kind of approach to working in Cuba, there was a lot more unknowns, and that was fascinating and exciting. In 2014 and 2015, as relations with Cuba started to kind of open and improve, a sister sanctuaries agreement was developed between Flower Garden Banks and Florida Keys National Marine Sanctuary and 2 marine sanctuaries in Cuba, and that kind of gave us the impetus and the opportunity to go and start to develop a relationship with the University of Havana and other partners throughout Cuba as well. We conducted a joint research cruise that was essentially half US scientists and half Cuba scientists, and it was incredibly successful. We were able to completely circumnavigate the Cuban archipelago. We went to 43 different sites, and essentially all of them, except maybe one, wasn't great. We found extensive Mesophotic reef habitat. And we think that this likely represents the largest known extent of Mesophotic reef habitat that's been described in the Caribbean to date. By the end of that mission, we had 4 new proposed MPAs just getting off of the ship, and 2 of those have been since enacted. And I'll talk more about it later, but we also were able to sample some corals and develop some molecular evidence of the kind of connectivity we might expect to see between Cuba, just 90 miles away, and Florida. And I'll talk about some of the implications for management and coral population persistence that come from those data in just a bit. So here's an idea of what the Mesophotic reefs look like in Cuba. A lot of kind of sheer walls and with a slope of maybe 80, 90%. Some areas of lots of branching, soft corals, other areas with lots of sponges or stony coral species, but essentially nothing bare. Lots of high diversity, high biomass, and strong evidence of good overall production from the benthic biota in that range. We did see some evidence of overfishing, lack of large grouper species, lack of large shark species, for example. But again, when you're working primarily with an ROV, we know that that's not always an accurate representation of all the fish that may be present. Stunning, absolutely stunning Mesophotic habitat. The next place that we started working at, or excuse me, the other place I wanted to focus on is Pulley Ridge. So Pulley Ridge is this kind of unique area that's on an extended continental shelf edge out from the dry Tortugas. It's an area that's been heavily utilized as a fish resource for several generations. And when we first started doing work there, we partnered with the University of Miami. We were supported by both OER and NCOS. And the goal was to try to understand whether or not Pulley Ridge was an important connectivity source for downstream areas in the Florida Keys, particularly the dry tortugas. The first thing we found after doing extensive surveys in this region was that Pulley Ridge had taken a huge hit. When we compared the data and the imagery that we collected to data and imagery from USGS earlier in the aughts, we saw an estimated 90% decline in coral cover and coral abundance in the region. That's within the original coral HAPC of Pulley Ridge that's indicated there on the far right. But what we also were able to document was an unknown, or at least not well described, coral reef resource west of Pulley Ridge in an area that was not currently protected really by anything. Again, led by John Reed, he proposed expansion of the Pulley Ridge habitat area of particular concern, and those additional coral reef resources were protected by HAPC designation in 2020. But we didn't want to stop there, and we started to take some of the same techniques that we'd used in Pulley Ridge and some areas of the dry tortugas. We went back for several successive cruises in the Florida Keys National Marine Sanctuary as well. And there we were really focused on trying to find and characterize these kind of not well-known mesophotic habitats, and we found several of them. We found an area off of Key Largo where just a few weeks before, Chris Taylor and colleagues at NCOS have been able to do some multi-beam mapping for us. They sent us back targets that said, hey, look at these bumps, you need to go check them out. And when we did, it was coral everywhere, at least 8 kind of almost contiguous kilometers of coral reef habitat at about 80 to 90 meters off of Key Largo. But we're certainly not the first ones that know about the importance of this habitat, because there was evidence of fishing gear all over the place, including wire bandit reels that had completely sliced off large, giant red barrel sponges. So it's an area that is an important resource, and I'm really glad that we were able to document that, as well as document that with the exception of those fisheries impacts, most of the mesophotic reefs there look remarkably healthy. Many of you are probably aware that throughout Florida and now spread throughout the Caribbean, we've been dealing with a new disease outbreak since 2014 called stony coral tissue loss. And it seems relatively absent from mesophotic reefs throughout the Florida Keys. That's not true of all mesophotic reefs. It has been documented in places like the US Virgin Islands. And we're concerned that it may be showing up in flower garden banks, although we're not 100% convinced of that yet. But at least here in the Florida Keys, there was not a huge impact of this disease in these deeper habitats. And so once we identified those habitats and realized how important and healthy they were relative to the shallow areas, we shared all of that information with Florida Keys National Marine Sanctuary. And not only have they added those additional mesophotic areas adjacent to the existing sanctuary, they've decided that Pulley Ridge HAPC should be part of the Florida Keys National Marine Sanctuary as well. So there's a proposed rule that's currently under evaluation, the Florida Keys Blueprint for Restoration, and that is open for public comment and scoping. And we're really hoping that, that all of you that are interested in advancing coral conservation will contribute your voices, if you're capable, to that process so that we can see those areas protected moving forward. In addition to benthic characterization and sampling for biodiversity in all of these locations, we also had this great opportunity to look at something I'm really interested in, which is coral metapopulation connectivity and persistence on a really big scale. So we started with one coral species, and that slowly kind of expanded to 2 and then 3 and then 4, particularly when we got an additional award through NCOS to work with Lehigh University on the Coral Connectivity Project in the Northwest Gulf. So as a result, we now have, for several of these locations, samples of these corals from multiple different depth ranges, that allow us to look at both their population genetics and their algal symbionts to look for differences. I'm not going to talk about the algal symbionts today in terms of time, but I did want to talk a bit about some of the coral connectivity results that we found. So for the most important striking message that we found is when we optimized this 2B rad single nucleotide polymorphism pipeline, essentially a DNA fingerprinting technique. What we found for corals that are relatively ubiquitous, like Montastria cavernosa, is that there is strong evidence of connectivity across broad ranges and across depths. So what you're looking at here in this chart are several populations of both shallow and mesophotic Montastria cavernosa. The up arrows associated with each of the diagrams denotes the shallow population. The downward facing arrows on the right side of each chart designate the mesophotic population. And the different colors within those populations represent the relative potential admixture of 4 different identified lineages or populations within this species in this range. And each individual column represents an individual coral colony. And so what you find is that in places like the Northwest Gulf, they're really well mixed. There is no difference in the population structure between the shallow and the mesophotic habitats. And we can then expect to manage them as one contiguous unit. But in other locations like Belize or like the Upper Florida Keys or like off the northern coast of the Yucatan, there are some really strong differences between the shallow and mesophotic populations, indicating a potential lack of gene flow between the two. But that doesn't mean they're necessarily isolated. We've also found evidence of gene flow from Mesoamerican mesophotic reefs towards Cuba and all the way up into South Florida as well. So even if they're not mixing with their adjacent shallow habitat, they may be providing larvae over a series of generations that allows them to connect to shallower habitats downstream. And recognizing these modes of connectivity and these metapopulation dynamics that allow for corals to persist in places like the Florida Keys suggests that if we're going to have a robust infrastructure that allows for coral reproduction into the future, almost by default, we're going to need an international management network to support population persistence, working with partners in Cuba, Mesoamerica, and Mexico. And that may mean, for example, that, equally important to protecting the habitat resources we have here, we maybe need to invest in population persistence in these areas because they're providing important larvae to us downstream. When we looked at another coral species, specifically in the Florida Keys, Stephanocini intercepta, this is work from a grad student of mine, Ryan Eckerd, who used to work for Flag Garden Banks for many years. We found some interesting patterns that suggest that not only might these mesophotic populations be healthier overall, but there's evidence of essentially reduced diversity within the shallow populations. They appear more inbred in the shallow areas in the Florida Keys, and there's a higher amount of overall relatedness, suggesting some kind of either sweepstakes reproduction that has led to a condensing of that population, or losses of many individuals, perhaps as a result of disease. But nonetheless, what it means is that we have relatively more diverse mesophotic populations for this species in the Keys across all the sites we sampled. And what was really interesting, here I'm showing the relative estimated amount of potential larval supply from each of these sites. And this is the shallow supply or larvae that theoretically could be coming and gene flow that could be coming from the shallow areas. Here's mesophotic. And so what you see is that overall, there's a much stronger likelihood that the mesophotic populations are important larval sources for this species in the Florida Keys. And this may be true for other species as well. So when it comes to managing this species and identifying areas that need to be protected, these mesophotic regions within the Keys are particularly important. Especially areas that we hope will now be included in the expansion just off of Tortugas Ecological Reserve West. That's an area that is not currently protected, but is proposed to be. So let's move on then to talk about some of the work that we've done, particularly with sponges in terms of new methodology and techniques and potential applications for pharmaceuticals. So a lot of this work has been led by Shirley's team, along with her student Megan Conkling, and partnering with Amy Wright and Esther Guzman here at FAU Harbor Branch. And they've had some really important discoveries and method developments here. The first is that we believe they've created the first, and to our knowledge, only biobank of cryopreserved sponge cells, which is essentially allow you to have a repository of diversity. And then it becomes a source for cell culture. And cell culturing in invertebrates is notoriously difficult. We've got it pretty well dialed in for humans, vertebrates in general. Think of all the different human cell lines that can be tested. But this was never before achieved in invertebrates until Shirley's team was able to do it. So they optimized some conditions to establish the very first invertebrate cell line, and it happens to be a sponge. And it creates a whole new experimental system to try to understand how sponges are behaving, what controls their biological functions. And it also potentially creates this availability to scale up the amount of production. And if we start thinking about potentially having to do sponge restoration, there could be opportunities to do that both with farming individual sponges, as well as developing cultures of totipotent cells that can become adult living sponges as well. So because sponges are essentially trapped on the benthos, for many organisms that are, they ultimately end up competing for space. Sponges in particular, because they're filtering water all the time, are constantly being exposed to and invaded by potential new bacteria, pathogens, etc. So we see that there's an increase in the likelihood of sponges or sponge-associated biota, things that live in and on sponges, to develop secondary metabolites, things that can combat invaders or combat microbes from growing on their tissues, or at least unfavorable microbes. One of the examples of this is from a sponge called Afrocolistin. So an extract from this sponge has shown really strong activity against cancer cells, particularly against protecting against the mutations that can occur in DNA repair. And this is a really interesting finding, because there are certain kinds of cancer, in particular triple negative breast cancer, that have almost no treatments to date. And so having a very selective compound that can go in and target the problems in mutation rates could be a really important treatment for this, this devastating disease. Another one that has shown a lot of promise is what we lovingly call the Shrek sponge there on the bottom right. This was isolated from a West Florida shelf expedition that we did. And this one is near and dear to my heart because it shows strong activity against Alzheimer's. And so for anyone who's ever had interactions with friends or family that have dealt with Alzheimer's, you know how devastating that disease can be and how limited the treatment options are as well. So we're hopeful about both of those and their prospects for the future. Moving on to some of the exploration tech development that we focused on. One of the first things that we did really early on is that we knew that subs are expensive to operate. And we started to think about how could we do the same kind of things that we've done with the Harbor Branch Johnson Sea submersibles in a more nimble, cheap package. And so essentially what we did is we tapped into the team of engineers that have worked on the Johnson Sea submersible and said, can you build us a package that can go on the bottom of an ROV that gives us some of the capabilities that the JSL has, but on a smaller scale? And they knocked it out of the park. They developed a package that quickly bolted on to the bottom of the UNCW Mohawk ROV that allows us to easily collect sponges, with a little bit more difficulty, corals, but algae, soft corals, all of those are really quick and easy to sample either with the manipulator or with a suction rosette that goes into the back as well. So we've made extensive use of this capability on all of these expeditions that I spoke about earlier. But ideally, we don't have to take a piece of coral that may have taken decades to grow every time we want to know something about it. And so, inspired by the idea to get non-destructive information from corals about their relative health and metabolism, Alina Schmant's team at UNCW started down this really successful path to come up with a new instrument that would allow us to measure coral in situ metabolism on the fly by a diver in a non-destructive way. And so they developed KISME, or KISME, as she likes to call it, which is the Coral In Situ Metabolism Instrument. And it's been successfully deployed in several different areas. And it's been so successful that this has now gone to full commercial application. So they had several prototype phases, and now you can go buy one from Qubit if you are interested in this kind of capability. But in some cases, just getting physiological data is not sufficient. You need a tissue sample. And we recognize that the smaller the tissue samples we can take, the less our potential impact on those organisms. And in many cases, we're improving our ability with the molecular techniques so quickly that we only need a very small amount of tissue to answer the questions that we would like to answer. So to focus on trying to develop a capability to get these smaller samples, at least with the diver and potentially in an automated fashion in the future, we embarked on a project that was called the Stinger with the Institute for Human and Machine Cognition, led up by David Fries. And so there's a lot of back and forth from those of us that are in the field collecting samples on a regular basis, and this group that kind of was thinking outside the box on how we could create a quick puncher that would instantly collect the sample and potentially preserve it at the same time. We also had quite a bit of help from some of the staff here. Shirley and Don both contributing quite a bit into helping to refine this instrument in a way that could be more successful. And it was deployed in one of the NEMO missions at Aquarius as well. Not without problems, but with relative success. A platform that has found far more success overall has been our integrated Midwater exploration platform. Many of you are aware that there's been an increasing interest in mesopelagic exploration and characterization. And we have several optical specialists, kind of marine optical engineers that couple with biologists in our programs. This has been predominantly led by Mike Twardowski, and using a combination of both OER support and ONR, Office of Naval Research support, they developed this platform that really took a whole bunch of different technologies, including some that have been developed here, and put them all on one platform that could all be deployed at one time to look for verification of data across different sensing techniques, as well as a more holistic integrated approach. So this combines a splat cam, in other words, something bioluminescent hitting on it, creating a bioluminescent profile, acoustic fish profiler, zooplankton profilers, LiDAR technology, and a bunch of the standard sensors you would expect for oceanography as well. And this has become a great test bed that has been deployed several times to develop technological assets through our Cooperative Institute. And then lastly, on the tech front, we had 3 different teams that all recognized this bottleneck that we all get into when we go out and do either ROV surveys or even diver surveys, where we come back with thousands and thousands of images, and the relative amount of manpower, human power required to analyze those is immense. And so developing capabilities to automate that in a way that's reproducible and reliable was really important to us. We had 3 different teams they kind of by design, we had independently working on this and then comparing the relative approaches and outcomes. And some of the techniques that they found that have been most successful so far have been to develop these convolutional neural networks, aka machine learning, that allows them to iteratively go back and improve this over and over again. And we've used some training sets from places like Pulley Ridge, where the benthos is not too crazy diverse. It has some great success. And then really the potential benefit that would allow a translation of this into research labs is making it so that not everyone has to necessarily be a programmer in order to use it. So they've also developed a number of graphic user interfaces that automate some of these functions as well. This is mostly work that's been led by Bingyu Yang, Jose Principe, and earlier Dylan McNamara as well. So then lastly, I wanted to wrap up talking a bit about our impact in terms of education and some of the future directions that we have planned as well. So one of the things that we really jumped on early on was we all can point to periods in our careers where if we got out to sea or if we got to a research station, it was completely transformative. And we really think that's a valuable thing for people to do. Telepresence is bringing that initial introduction to more and more people. I'll talk more about that in a minute. But getting people out to sea was still a big important goal for us. And so we developed these ocean discovery cruises with support from not only our Cooperative Institute and OER, but also support from Florida Institute of Oceanography and our foundation here at Harbor Branch. And essentially each spring we sent groups of anywhere from 16 or so undergraduates at a time in 2 blocks of 8 usually out on these missions. And to date, we've had over 100 students that have been able to get out to sea, sample and go through things with their own 2 hands and their own eyes. But we also recognize that that's still a very small number. And if we want to get even more exposure to more people, telepresence is going to be a tool that enables us to do that. So through our Cooperative Institute, we established a command center here on the Harbor Branch campus. Special thanks to Casey Cantwell, who came down and helped us to optimize that and think about our workflows and how that can work most effectively. And so far, we've contributed to, that number's a little out of date now, 24 NOAA telepresence missions. And on 6 of those, we've had CIOERT personnel, either Stephanie Farrington or myself, as science leads on those missions. We also developed a whole new graduate course based entirely around the idea of telepresence. And then to date, we've had over 400 student participants and 300 public visitors to our ECC. We have a high school here on campus that's marine science focused, and many of those high schoolers have gotten to participate and interact with us when we're out at sea as well. A few additional metrics of our overall success. To date, we have 167 peer-reviewed publications and counting. There are several more that are in review, and we know that for the next several years, more will come from work that was funded by OER. We've presented at more than 230 conferences or submitted technical reports from those. One of the things I'm most proud of is that we've had 139 direct student participants in CIOERT research. That's either graduate or undergraduate thesis researchers or summer interns whose projects were specifically focused on a CIOERT-related task. We also developed a new marine science and oceanography graduate program here at FAU. It's not only at Harbor Branch, it's across the entire university, and it's been really one of our growing and successful programs. And then again, through our local high school, we've had several folks that have been able to come and participate in our ECC work. And then we also developed a coral genomics-based module for the local high schools to be taught. And that's been taught to several hundred children as well. So a couple big kind of conclusions, overall outcomes. Again, if nothing else, I want you to know that really, Mesophotic reefs are fascinating. They're extensive. They're relatively well connected across this broad area of Florida, Gulf of Mexico, Cuba, Mesoamerica. And because they're more robust, we're hoping that they may help to facilitate the recovery of some shallow reefs. This idea of them being a refuge from some of the maladies and challenges that shallow reefs are facing. We've been able to achieve several new MPAs. We've been able to help provide data and information to support sanctuary expansions and to revise management plans across several different regions. We've developed several novel biotechnology applications for marine sponges, again, including that first invertebrate cell line and some promising treatments for some human diseases. Through a kind of disciplined innovation approach, we've been able to give new techniques or new machines that allow us to explore oceans in a different way, to sample them in a different way, or to understand them in a way that we haven't before. And I think that's one area that there's still a lot of room for growth. And then lastly, providing programs and opportunities to inspire and develop the expertise in that next generation of ocean explorers has been really important for us too. In terms of where we're going from here, although our Cooperative Institute is sunsetting, we have several existing and ongoing NOAA projects that we're still going to be participating in. So we're part of that NCOS cycle project in the Northwest Gulf. We also have an award from OER that's focused on blue economy biotech. And there was a cruise that was just completed earlier this year focused on that. We've also received support from both NOAA and the Florida Department of Environmental Protection to expand this integrated connectivity approach and to use several different OMIC techniques to look at evidence of connectivity across large and mixed spatial scales. We also remain a member of CMIS. So although this Cooperative Institute will be ending, we are still actively involved with CMIS and can still route projects through CMIS and partners through CMIS in the same way that we used to through CIOERT as well. And then recently we established a new center here at Harbor Branch, the Center for Coastal Oceans and Human Health. And this is another area that we see really expanding in our region going forward. And we know that there's going to be several opportunities, not the least of which is a recent call from NIH and NSF. So again, thank you all. If you would like to contact me, here's some information for you. And here's a hopeful, inspiring video of some of the first records of mesophotic coral spawning ever recorded. And with that, I'd be happy to take a few questions. Thanks, everybody.
Thank you for your presentation, Dr. Voss. This is really, really interesting. I love the footage in particular. To the audience, we have about 7 minutes to take your questions. So please type them into the questions chat box and I'll read them to Dr. Voss. And while we wait for some questions, I just want to let you know that we are recording right now, and we will be uploading the recording to the NOAA Central Library YouTube channel in the next few hours. So if you'd like to share this recording, we encourage you to visit YouTube to access it. I do have a question. This first question asks, have you thought about investigating mesophotic reefs off the exumas? I don't know if I'm pronouncing that correctly.
Off the exumas. So I would love to know who that question came from. I can't click on that right now. So I did all my graduate work out at Lee Stocking Island. In fact, that's where I met my wife as well. So the exumas are near and dear to my heart. Earlier, when we were younger and crazier, we did some kind of, I'll just say, probably not very smart bounce dives on air into the Mesophotic zone. But no, we have not had the opportunity to go and do a lot of work in the exumas now, and it's an area I would love to expand to. We know it's there. It's undoubted that those are important and extensive Mesophotic habitats. Some of them have been mapped a little bit with some earlier work that Harbor Branch did with the JSL in the 80s and early 90s, but not extensively in the exumas, and there's certainly room for expansion there. And if someone who asked that is keen to collaborate, give me a shout.
Okay. I will be sending all these questions off to Dr. Voss after the presentation, so they'll let you know. you got a compliment from somebody in the audience who said, excellent presentation, Josh. Thank you so much. And it's going to give it another minute to see if we get any more questions. I'm, of course, very interested in the Shrek coral that you mentioned and the potential of that. That was very exciting to see. Give it another minute. Oh, another appreciation. Great job, Josh. Appreciating all the work there.
We appreciate the support. A lot of folks I'm seeing in the audience are people that have helped us to achieve these things and couldn't do it without them. Thank you all for joining today and for all your support for the past 13 years.
Absolutely. All right. Well, definitely contact Dr. Voss offline if you have a question that comes to you later on. Oh, one more compliment. Great to see the work of technical diving helping to achieve the missions from another person in the audience. And totally agree. I was fascinated by this whole presentation. I'm going to say thank you again to NOAA's Cooperative Institute for Ocean Exploration, Research and Technology for organizing this presentation, and in particular to you for sharing all the science with us, and to Dr. Mashkor Malik for introducing you. And with that, enjoy the rest of your day, everybody, and please let people know about the presentation. I think there's a lot that is here that a lot of people could enjoy and learn from. Have a wonderful day, everyone.
Thanks, Lisa.
Thank you.